All right, so for lecture 2, let's look at the physiology of vision. We will briefly go over the anatomy, but we will do the detailed anatomy in lab. So the human eye can see the visible light spectrum, and the visible light spectrum runs from about 350 to 750 nanometers, so it's a fairly narrow range of our wavelengths of light. And when the light does strike the eye, it is refracted. And so what refraction is how the light bends when it hits a particular medium. And so air has a very low refractive index, and so it doesn't really bend as it travels through air. But if it strikes something like a liquid, the refractory index is greater than 1, so it bends the light. And so that's what the eye is doing, as the light strikes the cornea and then moves through the lens. And so if light strikes a convex surface, the rays converge to a focal point. If it strikes a concave surface, then it diverges and spreads the light. And this is what your eyeglasses are actually doing when it's correcting your vision. It is adjusting the focal point. So depending on whether you're nearsighted or farsighted, it will focus or converge the light so that the light will strike the retina where it's supposed to. And so at a distance of less than 20 feet, light waves diverge sharply. And at distances greater than 20 feet, light rays are almost parallel. So it's finished diverging at that point. So if you look at an apple on a distant tree, the rays are almost parallel when they reach your eye because you're looking from far away. And the cornea must flatten to provide enough refraction to focus the light on the retina to make the light beams converge back to a single focal point. But when you look at an apple up close, then the rays are not as parallel, and so the eye needs to round the lens to provide the additional refraction needed to focus the rays on the retina. So with that light refraction, 2/3 of the refractive powers are in the cornea, that clear area in front of the eye. And then the lens fine-tunes the refraction to make it strike the retina at the correct spot. Now remember, with our acute color vision, with the fine-tuning, it's going to strike back on that macula densa where the fovea centralis is. That's where all of your cones are. And so here you can see that ciliary process. And so the ciliary body with the suspensory ligaments hooked to it hold the lens in place. And so smooth muscles of the ciliary body relax. And when they do that, it moves away from the lens, putting tension on the suspensory ligaments and flattening the lens. So pulling away. And then when you need accommodation, ciliary body contracts and the lens rounds up for near vision. And so let's flip back and forth so you can see how it's flattened when it relaxes, and then it is rounded up when it contracts for the near vision. And so that contracted ciliary body moves closer to the lens, removing the tension on the suspensory ligaments and allowing the lens to round up. So that is how the fine-tuning of the lens is accomplished, via the ciliary smooth muscles and the suspensory ligaments. And so a normal eyeball, an emmetropic eyeball, strikes just on the retina, but some eyeballs are of different shapes. And so when they are of different shapes, you need lenses to help focus that point. And so a hyperopia, a farsightedness, the eyeball is too short. And so to correct too short eyeball, then you're going to go use a convex lens to get the focal point to pull forward. And then if your eyeball is too long, a myopia for nearsightedness, then you're going to use a concave lens that diverges the light so that it strikes the retina further back. And that gives you the clear vision with your glasses. And so what is the actual physiology of the light? And so here is the structure of the retina. Remember, we have the three layers. We have the outer fibrous layer. We have the outer fibrous layer made up of our cornea and the sclera. Then we have the middle vascular layer. Majority of that is this choroid. And then we have the neural layer that sits against the choroid. And the front of the vascular layer is the iris and the ciliary body. And with the retina, the neural layer of the eye, you have the pigmented layer that helps to keep the light from scattering. And then you have the rods and the cones embedded in that pigmented layer of the retina. So the light enters the eye. Here you can see the light entering the eye. It travels all the way through to strike on these discs of the rods and the cones. And here's another visual of it, kind of more how the light comes in. So the light strikes, travels all the way through, and then strikes these discs at the base of the rods and the cones. And so when the light strikes those discs, it actually causes a hyperpolarization in our rods and our cones. And so normally the rods and the cones are actually firing an action potential, but it's an inhibitory action potential. So it stops the action potential for traveling into the bipolar cells. What stops the bipolar, it hyperpolarizes the bipolar cells. And so it's an inhibitory signal to the bipolar cells. And then when you activate the rods and the cones, it hyperpolarizes those and they stop inhibiting the bipolar cells. And then the bipolar cells can now fire action potentials, then send it to the retinal ganglion and then out of the eye back to the occipital lobe to be interpreted as sight. And so here is a close-up of our rods and our cones, the discs that are in the rods and the cones and embedded in the cell membranes. So this is our phospholipid bilayer. Embedded in the cell membranes is the protein rhodopsin. And so rhodopsin is made up of opsin, which is this large bluer molecule, and cisretinol, which is this guy with his tail folded. And so an active rhodopsin has the cisretinol with the folded tail. And then when light strikes within that visible light spectrum between 350 and 750, it strikes the rhodopsin and causes it to dissociate. And so the tail of the cisretinol now converts to transretinal, straightens out, and it dissociates from the opsin. And that is what activates. And we'll see that in just a minute. And so cones contain a picnic, not the rhodopsin. So the rods have the rhodopsin. The cones actually have something very similar called idoopsin, and composed of retinol and the protein photospin. And so photospin, similar to opsin, photoopsin, similar to opsin, has a slightly altered structure which allows it to absorb the red, blue, and green that our cones see. So you have blue cones, green cones, and red cones. And so those three forms of the photopsin allow responses to different wavelengths. And so all the vast array of colors that we see are interpreted by striking the red, green, and blue cones and putting that together into all the different hues of color that we do see. And so here is in the dark. So that's why it's in this dark field. And so when it's not activated, light is not striking it. So the photons are light are not striking these discs. And so here is the plasma membrane of one of those individual discs in here, the rhodopsin embedded in it, or the idopsin, if it was a cone. And you have the cisretinal, it's inactive. And like I said, when it is inactive, the gates are actually open. And so this is constantly being depolarized, and that signal is inhibiting the bipolar cells. So in the absence of stimulation, photoreceptors are depolarized and continuously release neurotransmitter, and that inhibits the bipolar cells so that they can't fire an action potential. Now, when light strikes, the light striking causes the cisretinal to convert into transretinal. This causes it to dissociate from the opsin, and the secondary messenger, the metabotropic signal, now closes the ion gate. And so now that the ion gate is closed, it hyperpolarizes. So the closing by that secondary messenger signal stops the sodium from flowing in and causes the disc to now hyperpolarize. And then once it is hyperpolarized, let's go on to the, and so now once it is hyperpolarized, as I mentioned before, so normally when it's depolarized constantly, it is inhibiting the bipolar cell. But now that we have the cisretinal into transretinal and we are hyperpolarized in the rods or the cones, now that inhibitory signal has stopped, the bipolar cell can now send a signal, and that is what gives us our sight. And you can see the firing rate here. So in the dark, firing rate is very minimal, but as soon as you stop inhibiting the bipolar cell, and it can now signal the retinal ganglion cell, you get a firing rate this quickly. And so now we have sight and what we see. And so to come back to here, at transretinal is stays dissociated for a while, and it takes it a little bit to come back to its original shape. So rhodopsin is bleached when transretinal dissociates from opsin and can no longer respond to light, and so we have the bleaching effect. And then once you allow for adjustments, the light is suddenly reduced, cones can no longer function, rods are slow to regenerate, so it takes it a while for that cisretinal to reform and go back. And so it can take up to 40 minutes for rods to completely become functional, and the retina is extremely sensitive. And so with the light, when the light is suddenly increased, bleaches those rods and cones, resulting in a blinding glare. However, cones can regenerate functional pigments faster than the rods can. And so that's the accommodation. And a common defect, a genetic defect, is color blindness, and it's an X chromosome associated deficiency, and so that's why there are more males, because males only have the one X chromosome. Females can, but they'd have to come from a colorblind father and a carrier female or an affected female. So 8 to 10% of males have some form of color blindness compared to fewer than 1% of females, because of the fact that they normally have at least one normal copy on the X chromosome. And so what happens is you have a mutation in either the blue pigment or other pigments, but blue is the most common, and so then they can't see the reds and the greens. And so if you look at this color palette, you should be able to see the number 74. But if you can't see the number 74, then that means these are the Ishura plates. Then that means you have some form of color blindness. And so there's a lot of these to determine whether you have red-green color blindness or blue-red, or all the different combinations of what you can have as far as color blindness. All right, so now we get the signal. It is traveling out of the eyes. And so the left side of the left eye comes and stays on the left side of the brain, goes back to the occipital lobe. But the right actually crisscrosses at the optic chiasm and goes to the right side of the occipital lobe. And same thing, the right eye, the medial side of the retina goes across to the opposite side, whereas the lateral side of the retina stays on the right side. And so this and the overlap of this is what gives us binocular vision, so depth. So depth perception is needed. Both fields of vision are needed for that depth perception. And so they come through the optic chiasm and then come back to the occipital lobe where the primary visual cortex is located. And then here's a close-up of that and how they are each carried to the different sides of the primary visual cortex in the occipital lobe. And so here is that stereoscopic vision, the binocular vision that gives us our depth perception. So if you lose sight in one eye, you do start having problems with curbs and things like that because you don't have the depth perception that you had when you had both vision in both eyes. So here is our big picture of vision. So the cornea and flattened lens provide enough refraction to get the light to strike where it should on the retina. Once it strikes the retina, we hyperpolarize the rods or the cones. Once it's hyperpolarized, now the bipolars can send the signal to the ganglion, then the ganglion carries that back to the occipital lobe, and we get a conscious awareness of that initial processing of shape, color, and movement in each hemisphere of that visual field to give us the complete picture of what is going on in front of us. So that is our physiology of sight, and we will have a third lecture on the anatomy of the ear. Let me know if you have any questions.